The natural environment that budding yeast evolved to inhabit can be rich and abundant, but it can also be a place of deprivation where essential nutrients are scarce or non-existent. Under such stressful conditions, yeast develop into spores, which are long-lived and durable gametes. Yeast sporulation couples the scrambling of genetic information through meiosis with the packaging of the resulting meiotic nuclei. In my lab in the Department of Molecular Genetics at the University of Toronto, we use the sporulation program to understand fundamental aspects of development. In the course of our study as a graduate student in the lab, Michael Eastwood made a fascinating observation. Using live cell fluorescent microscopy, Mike was following the 4 meiotic products and observed what their fate was during low-carbon sporulation. In a very short period of time, 2 of the 4 meiotic nuclei simply vanish. Now, work in Breck Bayer's lab many years ago found that under reduced carbon conditions, yeast cells execute meiosis normally, but only package 2 out of the 4 meiotic nuclei into spores. They hypothesized that this was an adaptive response that balanced spore number with spore fitness. More recently, research in the labs of Aaron Neiman and Michael Knopp showed that yeast do not package 2 spores under this condition merely because they run out of nutrient to fuel spore construction. Rather, this is a developmentally programmed decision. Up to this point, however, the fate of the meiotic nuclei that did not develop into spores has not been investigated. When we saw that these discarded nuclei vanished rather acutely, we immediately thought of programmed cell death mechanisms. What we describe in our paper is that the unpackaged nuclei are actively destroyed through complementary cell death-related pathways. In the first pathway, the protein components of nuclei are destroyed through an autophagic pathway which most closely resembles mega-autophagy, a much less well-studied autophagic mode. During mega-autophagy, the vacuole becomes permeable, releasing hydrolytic enzymes that consume the cell. Mega-autophagy is an autophagic pathway that has been described in plants and is quite similar to mammalian lysosomal membrane permeabilization. What we found is that following the completion of meiosis, the vacuoles appear to become permeable and then blow up. We next evaluated the other principal component of nuclei, the DNA. Mike found that during programmed nuclear destruction, DNA was fragmented into nucleosomal ladders of approximately 150 base pair periodicity, a hallmark of mammalian apoptosis.
Through the course of studying the cell biology of programmed nuclear destruction, sporulation, we utilized liquid sporulation protocols, which are almost certainly not representative of what yeast would encounter in their natural environment. To ask whether PND is something that they actually utilize, we looked at wild isolates, which normally possess a very robust sporulation program. We examined them as they grow in colonies, probably the best circuit we can find to what they actually do naturally, and found that not only did they sporulate, but they sporulate to a reduced spore number and robustly execute PND. This suggests that PND is indeed probably a very useful and routine aspect of the yeast's natural life cycle.
Ultimately, our work reveals a new aspect of the yeast life cycle that involves developmentally programmed convergence of complementary cell death-related pathways. In addition to opening up the studies of programmed cell death mechanisms to the powerful tools of yeast genetics, our work has fascinating implications concerning the evolution of these programmed cell death mechanisms.
I'm Dr. Anna Spagnoli. I'm a professor of pediatrics at University of North Carolina School of Medicine, and I'm going to talk with you today about some exciting data that we found in my laboratory. Osteoarthritis is the leading cause of disability in adult population in industrialized countries. So there is no form of treatment for osteoarthritis, and the therapies are primarily focused on controlling the pain of patients with osteoarthritis. So we don't have any therapy that is able to prevent the disease, delay the onset, or halt the progression of the arthritic process. Despite of the clinical significance of joint development, we know very little on how joints develop. And understanding joint development can lead us to understand how joint degenerates in the arthritic process and potentially to target new therapeutic pathways to treat the osteoarthritic process. So in our study we use very elegant genetic manipulations as well as pharmacological approaches and cutting-edge techniques such as laser capture microscopy to isolate the joint progenitors and to gene profiling the environment in which joints developed. And surprisingly, we found that joints actually develop in an environment that is characterized by dramatic downregulation of cytokines. The same cytokines have been known to be involved in the osteoarthritic process, but our studies show for the first time that the same cytokines are actually involved in the developmental joints. We have provided unequivocal evidence that MCP5, that is downstream of TGF-beta type 2 receptor, regulates the joint's development. In particular, we found that it is a negative regulator of joint development while it's controlling the progression of the chondrocytes in the growth plate. Our studies are summarized in Figure 1. As shown in the upper part of this figure, in the presence of TGF-beta type 2 receptor, the MCP5 within the developing joints is downregulated. And this downregulation allows the formation of the joints. And the same downregulation also allows in the growth plate that you can see in the bottom part of the figure to progress to hypertrophy. In the absence of TGF-beta type 2 receptor as shown in the next figure, the MCP5 becomes upregulated, as you can see on the upper part of the panel. And the upregulation of MCP5 does not allow the formation of the joints while the growth plate becomes completely disorganized. Our study is important for several reasons. First, it provides new important insight on the mechanism that govern joint development. It expands our understanding on the role of TGF-beta in joint cell fate and differentiation. It identifies novel therapeutic target of the TGF-beta pathway, and it shows for the first time that the downregulation of cytokines such as MCP5 is essential for joint development and growth plate differentiation. Our studies really represent a paradigm shift in the field of joint biology and osteoarthritis. Until our study, joints were perceived as the passive victims of the inflammatory influx that will lead to the osteoarthritic process. The novelty of our study is the fact that we found that joints actually hold an active mechanism to repress cytokines, and this mechanism starts during the fetal life and is tightly regulated by the TGF-beta type 2 receptor that then downregulates the MCP5. So our finding really represents a milestone in the joint biology field because we can now target the TGF-beta type 2 receptor as well as the MCP5 as potential therapeutic target to empower this active mechanism to repress the joint degeneration in osteoarthritis.